Thank you very much. It's a great pleasure to be here this morning. The massive venture to reconfigure the genetic core of the world's food supply purports to be based on solid science, and its proponents accuse people who raise concerns of being ignorant of the relevant science or even of being anti-science. But in reality, this venture is not firmly grounded in science, and in order to advance and survive, it has routinely violated the basic principles and protocols of science. Moreover, this unpleasant reality has been obscured through systematic misrepresentation. Basic facts of biology have been habitually distorted. The process of creating genetically engineered foods has been deceptively described to make it appear far less disruptive and much more precise than it actually is. And false statements have consistently been issued about the tests on these foods to cover up numerous troubling results. Although these assertions may seem overly bold, my book provides solid evidence that fully backs them up. And the solidity of this evidence has been attested by many experts. For instance, John Eichert, Professor Emeritus of Agricultural Economics at the University of Missouri, has stated that the evidence is comprehensive and irrefutable. Moreover, the irrefutable fact that the facts have been routinely misrepresented is in itself concrete evidence of how strongly the aggregate evidence weighs against the scientific soundness of the GE food venture. Because if the evidence were truly supportive of the venture's soundness, there would have been no need to distort it. Furthermore, the unsoundness of this venture can be demonstrated through every other angle of analysis as well. For example, the basic assumptions on which it was founded and on which claims of its reliability and safety were based have been proven wrong. One of these foundational assumptions presumed that the genome is a simple linear system in which the action of a single gene will not significantly impact the others and won't disrupt their normal function. For instance, in 2007, a New York Times article by the veteran technology reporter Denise Caruso observed, "The presumption that genes operate independently has been institutionalized since 1976, when the first biotech company was founded. In fact, it is the economic and regulatory foundation on which the entire biotechnology industry is built." That was the slide that should have been up there, excuse me. That's the problem with technology. It sometimes acts unpredictably. One of the other main presumptions was the notion that genes aren't arranged in an organized manner, and that the sequence in which they occur is essentially unimportant. Together, these two presumptions supported the belief that a chunk of recombinant DNA could be wedged into a plant's genome without inducing disturbance. Because if the behavior of the native genes was largely uncoordinated and their arrangement was irrelevant, there would be no important patterns that could be perturbed. Accordingly, they engendered confidence in the precision of genetic engineering by implying that its results would be consistently predictable. However, these influential assumptions, which provided the ideological foundation for a colossal endeavor to transform agriculture, have been decisively discredited. Abundant evidence has demonstrated that there is a high degree of coordination between genes and that their arrangement is highly organized. And this evidence undermines the claims about the safety of genetic engineering. In that New York Times article, Denise Caruso asserted, "Evidence of a networked genome shatters the scientific basis for virtually every official risk assessment of today's commercial biotech products, from genetically engineered crops to pharmaceuticals. However, biotech proponents have largely ignored these troubling implications, and such disregard has been essential for maintaining faith in the GE food venture, because the more the genome functions as a tightly coordinated system, the more potentially disruptive and unpredictable are the interventions of the bioengineers. And these interventions do cause disruptions, substantial ones. Moreover, although such disruptions are not induced by pollen-based breeding, they occur during every phase of the genetic engineering process. The procedures through which cassettes of recombinant DNA are inserted into isolated plant cells induce widespread disruptions. The process through which these isolated cells are then developed into mature plants induces another set of disruptions, and the subsequent activities of the inserted DNA can create additional disturbances. Numerous scientists have cautioned about these hazards. For instance, an expert panel of the Royal Society of Canada concluded that the default prediction for every GE food should be that the genetic alteration has induced unintended and potentially harmful side effects, such as the creation of novel toxins or allergens that could be difficult to detect. Therefore, these Canadian experts emphasized the need for rigorous safety testing, and so have many other experts. But regulators have not required rigorous testing, and the testing that's been conducted has, for the most part, been deplorably deficient. Even so, many studies published in peer-reviewed journals have detected adverse effects to the animals that consume GE foods. However, the proponents of these products either ignore this research or they unjustly attack it, and they persist in falsely proclaiming that there's no evidence any GE food has ever caused harm. The degree to which the facts have been disregarded and distorted is disgraceful, and it clearly demonstrates how seriously science has been subverted. This truth has been vividly expressed by Dr. Michael Antonio, a molecular geneticist at King's College London School of Medicine. If the kind of detrimental effects seen in animals fed GE food were observed in a clinical setting, the product's use would be halted and further research instigated to determine the cause and to find solutions. However, what repeatedly happens in the case of GE food is that despite increasing evidence of serious adverse health test results, government and industry continue unabated with the development, endorsement, and marketing of these foods as if nothing has happened, to the point they even seem to ignore the results of their own research. Thus, even this short summary has revealed that from the standpoint of biological science, the GE food venture is significantly unsound. And when it's analyzed from the standpoint of computer science, the picture becomes even more troubling. Such an analysis is highly relevant because both genetic engineering and computer science are engaged in altering complex information systems. And computer science has learned a lot about the risks of making such alterations. Moreover, it has learned that these risks are inescapable. That's an extremely important fact with major implications for genetic engineering. Software engineers have learned that when the information systems they themselves create become large and complex, there's no way to alter them with complete precision. Even when the alteration is a small refinement designed to improve the system's performance, the mere process of revising it in such an ostensibly minuscule manner is very likely to disrupt one or more of its other parts. This is an amazing phenomenon. Software systems are designed to be linear, which means they're structured so that a specific operation only produces a specific result. Operation X should only produce Y. However, despite the programmer's best efforts, their systems transcend the structured limits and, to a significant degree, behave in a nonlinear manner. Their parts interact in ways that were not programmed and cannot be predicted, which means that Operation X will not only yield Y, but may also induce Q and Z. Consequently, to reduce the potential for unintended interactions, software designers separate components that shouldn't interact and try to insulate them from such interaction. What they try to avoid is creating code that resembles this. They want to avoid writing what they call spaghetti code, a program in which the components are complexly interconnected. What they instead aim to create is ravioli code. They try to design systems in which the components that aren't supposed to interact are as independent from one another as the portion of cheese and vegetables within separate packets of pasta. Yet, even though programmers have succeeded in designing systems that are far more analogous to a plate of ravioli than a mound of spaghetti, and have reduced the potential for unintended interactions, they have not been able to eliminate it. Such unwanted results continue to occur. Before examining how these risks are dealt with, let's compare the characteristics of human-designed information systems with those of bioinformation systems. Let's compare man-made software with nature's software. Okay, we know that human-designed systems are designed to be linear, and they function in a largely linear manner. In contrast, bioinformation systems are inherently nonlinear, and they function in a highly nonlinear manner. The various parts are intricately interconnected, and every action can create a wide range of effects, many of which cannot be predicted. Software engineers avoid creating spaghetti code. They minimize interaction between different components that are not supposed to interact. But bioinformations are the most extreme instances of spaghetti code, the ultimate spaghetti code. And even if a human being had been able to create them, he or she could not adequately comprehend their various interactions. Moreover, despite the substantial knowledge we humans have gained about the workings of bioinformation systems, the extent to which our understanding remains deficient should be profoundly humbling. In human-designed systems, the rules governing how the parts interact are clearly expressed in written form, but only a small fraction of the rules of bioinformation systems are known, and most of those pertain to the mechanics of gene expression. Yet, as numerous experts have emphasized, many of the most important rules don't operate at the level of the genes. For instance, Richard Stroman, who was a distinguished professor of cell biology at the University of California, Berkeley, asserted that the most important rules operate at a higher level, the level at which genes are organized into functional arrays. And he noted that this level of gene management is not confined within the DNA, but is coextensive with the cell itself. Moreover, he emphasized that dynamics operating at this level are different than those at the lower ones, and that the interactions are far more complex, to the extent they are ultimately transcalculational, which, as he noted, is a mathematical term for mind-boggling. Several other experts have also pointed out that bioinformation systems transcend DNA and ultimately extend throughout the entire organism, while also extending far beyond our comprehension. So, mind-made versus mind-boggling. The systems, in the case of human-designed information systems, the rules and dynamics are comprehended by those who make revisions. In the case of bioinformation systems, the rules are transcalculational, and revisions are made with scant comprehension of what the rules actually are. So in light of these enormous differences, it would be reasonable to think that genetic engineers should exercise far greater caution than do software engineers, but in reality, they exercise far less. Software engineers recognize the inescapable risks of altering complex information systems, and they deal with them responsibly. Accordingly, when they revise programs, they routinely conduct tests to detect whether any problems have been created. Moreover, there's a special class of software for which the testing is extremely rigorous. It's called life-critical software. It's software that can cause loss of life if it malfunctions. Examples are the programs that govern pacemakers and X-ray machines, and the ones that serve as airplane guidance systems. In the US and the EU, not only are such programs required to undergo strict safety testing before they're allowed on the market, they also must be rigorously tested any time they're revised, no matter how minor the revision. The regulators will not accept arguments that the revised program is substantially equivalent to the former version. Instead, there must be a demonstration of safety achieved via systematic, stringent testing. But there's a glaring contrast in how risks are handled when disruptive alterations are made to the biggest, most complex, and least understood information systems on our planet. The biotechnicians don't adequately acknowledge the risks. In fact, they largely deny them. And even the regulators have discounted them and assumed there's little need for rigorous testing. The United States Food and Drug Administration claims that GE foods are so safe they don't have to be tested at all. And although regulators in the EU and most other regions have required some testing, it's been minimal. For many years, GE foods were allowed on the market based solely on some superficial showing of substantial equivalence to their conventional counterparts, something that is never allowed in the case of life-critical software. Despite the fact that a toxic tomato could cause far more deaths than a malfunctioning X-ray machine. And although the EU has finally required safety testing with the whole food, the tests are still astoundingly lax by software standards. Even if GE foods were compelled to undergo the same testing that's mandated for new pharmaceutical drugs, it would still fall far short of the rigor that's required when changes are made to life-critical software programs. Moreover, as my book demonstrates, if the kind of rigor that's required in software testing were mandated for GE foods, the entire venture would collapse. Consequently, the venture is incurably risky. Thus, when scrutinized through the lens of computer science, the GE food venture is not only seriously unsound, it's outright reckless. But what about the new ways of manipulating genomes that are referred to by their promoters as genome editing? Will they be a safe alternative? The answer is a definite no. So-called genome editing with techniques such as CRISPR-Cas9 is not a precise procedure. From the standpoint of computer science, such techniques entail abnormal risks and must be rigorously tested. Why? Well, the techniques themselves can induce off-target effects. 2, they must be accompanied by additional processes that entail risks. First, of course, putting the editing mechanisms into the cells causes widespread disruptions because they're the same kinds of disruptions that occur when genetically engineered foods are produced. And B, the transformed cells must be grown into mature plants via tissue culture, which is a process that causes, again, disruptions. In fact, some scientists have referred to the extensive disruptions caused by tissue culture as genomic shock. Tissue culture induces a genomic shock throughout the entire system. So if we cut through the promotional propaganda that's been passed off as science, and we carefully examine genetic engineering in light of our best scientific knowledge, genetic engineering as it has been practiced up to now and as it will be practiced in the future, it becomes clear that the venture is incurably risky and that the risks are unacceptable, especially when safe, sustainable, and well-proven alternatives are readily available. Thank you.